Now I'm very excited and honored to introduce our speaker, Dr. Florian Krammer. Dr. Florian Krammer graduated from the University of Natural Resources and Life Sciences, Vienna, Austria in 2010. He received his postdoctoral training in the laboratory of Dr. Peter Pallis at the Icahn School of Medicine at Mount Sinai, New York. Working on hemagglutinin stock-based immunity and universal influenza virus vaccines. In 2014, he became an independent principal investigator and is currently the Mount Sinai professor of vaccinology at the Icahn School of Medicine at Mount Sinai. Dr. Krammer's work focuses on understanding the mechanisms of interactions between antibodies and a virus surface glycoproteins and on translating this work into novel, broadly protective vaccines and therapeutics. The main target is influenza virus, but he's also working on coronaviruses, flaviviruses, hantaviruses, filoviruses, and arenaviruses. He has published more than 300 papers on those topics. Since 2019, Dr. Krammer has served as principal investigator of the Sinai-Emory Multi-Institutional Collaborative Influenza Vaccine Innovation Center, which develops improved seasonal and universal influenza virus vaccines that induce long-lasting protection against drifted seasonal, zoonotic, and future pandemic influenza viruses. I started to collaborate with Dr. Krammer on influenza viruses early in my PhD program. I also got my fantastic 2-week training in Florence Lab in 2016 summer. That was a long time ago. So the series program, I really enjoy working with him and doing all the wonderful work on influenza virus before. So today, let's extend to the impactful and long-lasting topic virus SARS-CoV-2. So Dr. Florian Krammer's title is entitled as Antibody Responses to SARS-CoV-2 Infection and Vaccination. Thank you so much for joining us. Please take away.
Thank you very much for the invitation and for this very kind introduction. So a few things first. Apologies for my voice. I had my first respiratory tract infection since 2019. It was a rhinovirus. It was not SARS-CoV-2, but my voice is still a little bit weird. And the other thing that I need to do is I need to probably skip a few slides here and there because I heard we have to be out of here at 1:50. So don't. I'll have to go through a few sections a little bit quicker. So what I'll do today is I'll share initially a little bit of a story of what we did in New York initially at the onset of the pandemic. And then I'll go into some topics with data that we haven't published yet that we find very interesting related to SARS-CoV-2, both vaccination and infection. Just to start with my disclosures, the main disclosure is that our school has filed IP on antibody tests and vaccines for SARS-CoV-2. So very brief introduction. We all know this. In December 2019, the end of the year, China informed the WHO that there was an outbreak of pneumonia with unknown etiology that was reported in Wuhan in China. And initially, the idea was that these cases are clustering around this seafood market, which is not just the seafood market; they have all kinds of animals. And it seems that this was really the source for these infections, although there's still some open questions. And the rest is basically history, right? I just got these numbers online yesterday. We have now officially 688 million infections for SARS-CoV-2 worldwide. The real number is probably much, much higher. It's very likely that the vast majority of the world's population has been at least infected once. And officially, we have about 6.8 million deaths. And you see the epi curves there. You're very familiar with them. But basically, initially, we had very few diagnostic tools. So initially you see very few infections, although you see a lot of deaths here on the right side. And then we go through the different waves. And as you probably know, this large wave here is the BA1 Delta wave, which also shows here up as a relatively severe wave in terms of deaths globally. Those are the official numbers. If you look at estimates for the real death toll in terms of excess deaths, we are probably more closer in the range of 22 million COVID-19 deaths. This is getting close to what we have seen for the 1918 H1N1 pandemic, but at least the lower range of that, although we have to say that the population was much smaller back then, and so it's still much less impactful as compared to 1918, but probably much worse than the other respiratory virus pandemics that we had since then. So I became aware of this outbreak also in the end of December. When you work on flu like I do, you're always looking out for zoonotic infections. Many of these happen in Southeast Asia, and so you look for stories like that. I was convinced this is a flu infection, and this is a zoonotic event where avian influenza would have spilled over into humans. But of course, I was wrong. And a few days later, on January 11th, I think the sequence was released. And when we downloaded the sequence, became clear, to everybody who did that this is a beta coronavirus that's causing the outbreak. And that's very closely related to SARS-CoV-1. At that point, my lab hadn't worked much on coronaviruses, but we studied a lot antibody responses to glycoproteins of influenza virus. And so we decided that we wanted to get prepared just in case this would come to the US. We did a few things. We actually started to bank samples that we got from our hospital already in January that were negative for RSV and negative for flu. And in fact, when we later on looked, we found the virus already in January in New York City. But we also started to make reagents that would be needed to characterize immune responses. And because I'm interested in antibody responses to glycoproteins, and because we hypothetically know that antibodies to the spike protein of coronaviruses can be protective because they can neutralize the virus, we focused on making a lot of these constructs that allowed us to produce recombinant spike protein and recombinant receptor binding domain. And so we started to set up assays for that. Initially, we had a huge issue with doing that because we had the reagents, but we didn't have samples. I asked the CDC if they would share samples from that one infection in Washington State. They declined. They said they didn't have an IRB to do that. And so we got samples from Finland and Australia to set up our assays. And that was just in time. That was in February. And in the end of February, we had the first official case in New York State, right? Or in New York. At that point, we were already helping our Department of Pathology, which runs the CLIA lab, the micro lab for the hospital, to set up PCRs. And they were also interested in having a tool so that they could screen people for antibodies to SARS-CoV-2. What they had in mind was a plasma program where they could find people who already have neutralizing antibodies, ask them to donate plasma, and then treat severely infected patients. And so we moved our ELISA out of our research setup into the CLIA laboratory, and it actually became the first laboratory-developed antibody assay in the US that was approved by the FDA for using CLIA laboratories. And this is just shown here. So they screened hundreds and thousands of people. In the end, the convalescent plasma therapy didn't prove to be very efficient, but at that point in time, we didn't have much else. And so that was something important to set up. At least that's what our hospital felt. And the other point that I wanted to make, we had these reagents very early on. We wrote a protocol of how to make them. We wrote a protocol of how to run these ELISAs. And we freely shared plasmids and protocols in February and from February of 2020 on. Basically without asking for agreements and MTAs, which you need now when you typically share reagents. We shared material with more than 250 labs worldwide, and then we also deposited these things in depositories. I actually got in trouble with our tech transfer office because I just freely shared these things. But honestly, I would do it again. I think that's something that's important in a pandemic. You have to act, and then you can ask questions later. But we had now a tool in hand to actually start to look into immune responses. And that's what I'm really interested in, right? What the clinical laboratory was doing was important for the hospital, but we also wanted to answer questions. And if you remember, in the beginning of the pandemic, there was a lot of discussion about people losing their antibody responses within 8 weeks, which is kind of biologically almost impossible. Reinfections that happen right away. And in the end, it turned out that this was persistent RNA. Of course, we had reinfections in the end, but that's a very different story. And so we sat down, and with we, I mean, Viviana Simon, who is my partner in crime with most of these studies, and myself, and we said, okay, there are 3 questions that we would like to try to answer. In that respect. And the first one was, how long do antibodies to SARS-CoV-2 last? The second one was, how frequent or how good is the protection from reinfection? And the last one was, can we define a protective antibody level? And we completely failed with the last one, although there are nice studies out there that addressed that and that actually produced really good numbers from the Moderna trial, for example. But initially, we actually succeeded with addressing the first 2. And in order to study this, we set up a cohort study in our healthcare workers, which we called PARIS, or Protection Associated with Rapid Immunity to SARS-CoV-2. And the goal here was to enroll about 500 healthcare workers in the hospital. The idea was that they would have the highest risk of getting exposure, so we would see reinfections there if they happen. And the idea was to have 50% of the people in the cohort to be naive and the other 50% to be SARS-CoV-2 antibody positive already. And this was started in April of 2020, and unfortunately, we already had a lot of people who had been infected previously. And then we wanted to follow them over time, look at antibody kinetics, and also study the risk of reinfection. And so that's what we did. Initially, the study was only performed at Mount Sinai, but NIH, who funded the study, really liked the idea and they asked us to expand it. And there's actually a number of sites that are involved now, but I'm only going to talk about the data that our site produced. And so initially we started, and this was the easiest to do, we looked at antibody titers. And when I say antibody titers, it's just binding antibody titers measured by ELISA. And so this is shown here. What you see here is on the y-axis, the antibody titer measured by ELISA. On the x-axis, days since enrollment into the study. So a lot of these people went into the study about 2 months after their initial infection. And then we looked at the antibody titers, and there's actually not that much waning that we see. There might have been more in the beginning, but we don't have the samples for that. And we lose a little bit of resolution in the end because those were healthcare workers. And of course, they got vaccinated, meaning they dropped out of that analysis. We had about 5% seroreversion, meaning about 5% of people lost their detectable antibody titers, which is kind of very similar to other virus infections that we see. And we also looked at infections in that cohort in this period of time, and this is shown graphically here on the right side. The blue symbols are naive individuals. The orange ones are people who had antibodies at the onset at enrollment. And then the mixed symbols are people who are seroreverters who lost their antibody titers. And these circles indicate infections. And so of the 11 infections that we detected, 10 happened in people who were naive and 1 happened in a seroreverter after the antibody titer dropped to undetectable, which is a pretty strong signal. This is nothing new. This is basically very similar to a lot of other studies that have been published and have been conducted, including 2 very large UK studies that show that protection from reinfection is substantial. It's relatively high, comparable to vaccines. And this was the case for the pre-Delta period, for the post-Delta period. But then protection against reinfection with Omicron, of course, dropped quite a bit. And the reason for that might just be that neutralizing antibodies raised by infection with pre-Omicron variants were not very effective in neutralizing Omicron anymore. But I think initially this was a little bit, there was a little controversy about that in general, when people were told that infection wouldn't protect you from reinfection and you would need to get vaccinated. I believe that vaccination reinforces the protection that you have from natural infection, but I think we also need to be careful when we communicate science to the population, and we shouldn't say things just because we want them to get vaccinated. As I said, this was a healthcare worker cohort, so this gave us a really nice opportunity to now look what vaccination was doing in these two different populations. And before I go into that, I just wanted to contrast infection-induced and vaccine-induced immunity a little bit. I'm not going to say that one is better than the other, but they are different, right? And we need to keep that in mind when we think about immunity. Infection-induced immunity induces immune responses against all structural proteins of the virus, but also all non-structural proteins, right? There's a lot of open reading frames that the virus expresses in our cells, and you will get immune responses against those. In addition to that, the virus that infects you is likely a quasi-species. It's not monoclonal, so your immune system sees more diversity. And then you might have persistence of antigen for quite some time in your body, right? There's a study from Rockefeller University showing that they actually found virus antigen a few months after infection in the gut. And with natural infection, what we get is, of course, systemic immunity, but you also get mucosal immunity in the upper respiratory tract, because that's the site where the virus replicates as well. The downside here is that the immune responses are very variable, right? Some people have really good responses, some people have relatively low responses. Vaccine-induced immunity is different. At least in North America, this focuses on the spike protein only. There's no other components in the vaccine, and it's a consensus spike. There's no variability. And typically when you get these vaccines injected, you mount a very strong systemic immune response, but not necessarily a mucosal immune response, specifically not really in the upper respiratory tract. And that's an important difference. All right, so we had our two groups in the cohort, and then they started to get vaccinated. And this is shown here, right? So on the y-axis, again, you have antibody titers. On the x-axis, you have time from vaccination from the first shot, which is shown on day 0 here. And in orange, you have people who had been infected. And in blue are people who are naive at the time when they get the vaccine. And so for the orange group, when they get their first shot, their titers go up very strongly. Then they get the second shot and nothing happens. Then there's a waning phase, and then it seems there's a stabilization phase of the antibody titers. And we see initially different kinetics for naive individuals. You get your first shot, the titers go up. You get your second shot, the titers go up further, so there's a boost. But then the waning seems to be the same, and the stabilization seems to be the same, although at the lower level, right? So there are quantitative differences, and initially there are kinetic differences. What I wanted to point out here is this is something that can be explained with B-cell biology, this waning, right? So this initial peak here is made by plasmablasts, which are short-lived B cells that are in the periphery. After 10 days, they're gone. They make a ton of antibody, but they die. The antibody has a half-life of approximately 21 days for IgG1. So that will stick around for much longer than the cells, but you know, the titers go down. And then at some point, you get this stabilization or a much lower waning. And the antibody that you see here is made by a very different B-cell population. That's actually made by long-lived plasma cells in the bone marrow. Those cells can get as old as you are. And so that's why you see different kinetics here. And that is important to keep in mind. And the ratio of this initial response versus the long-lived response can be different with different vaccine platforms. The J&J vaccine looks very different. But this can also be alarming if you have this huge amount of antibody that drops. The other point that I wanted to make is that, again, you have this variability in infected individuals. And so it's really good, even if you had an infection, to get vaccinated, because what happens is you get this very homogeneous high titers if you get vaccinated after you had been infected. And that's super important. And the last point that I wanted to make here is, so we monitored infections during that period of time too. And before Omicron, we had 0 breakthrough infections in the orange group, and we had 14 in the blue group. And this could be a function of the magnitude of the response, but it could also be a function of the quality of the response. After Omicron, we started to see breakthrough infections in both groups, but the trend held. There's proportionally many more in the naive group, in the blue group as compared to the orange group. And so hypothetically, the idea is when you get a natural infection, and I touched upon that a little bit already, when you get a natural infection, you get induction of systemic immunity in the periphery that basically protects your lung very well. A lot of IgG is made, and that goes from serum into the lung. That protects you from severe disease. But you also stimulate a local mucosal immune response in the upper respiratory tract. It's mostly secretory IgA, and that hypothetically protects you from infection if the titers of neutralizing antibody on the mucosal surfaces is high enough. With vaccination, you skew that towards a systemic response that protects the lung very well. Some of the IgG might end up in the upper respiratory tract initially when you have these really high titers, but that's gone relatively quickly. And one idea to fix this issue, because you don't want to leave the upper respiratory tract unprotected, is intranasal vaccination, which can hypothetically induce these mucosal immune responses. And so we wanted to look into that in our cohort as well. And that's what you see here. Again, on the left side, you see the hybrid immune individuals. On the right side, you see the naive individuals. And what you see in the upper panel is IgG in the upper respiratory tract, in this case in the saliva. And what you see in the lower panel is secretory IgA. So that's that locally made antibody. When we look at saliva IgG, which comes from the serum, we basically see that in people with pre-existing immunity, so they have been infected, there's a huge induction of this saliva IgG after vaccination. And you also see that induction in people who were naive before vaccination. And again, this makes sense that there's a difference because we see that there is a titer difference in serum as well. When you then start to look at secretory IgA, you see that, first of all, you already have these types of antibodies before vaccination in the infected individuals, which makes sense. And then vaccination boosts those titers. And that was a surprise because we actually didn't assume that that would happen. And we don't understand the mechanism of it, but it does happen. It doesn't happen in all individuals, but in some of them, it happens very strongly. And you see very little of that in the naive only group. And so that is an interesting finding, but we think we need to find ways of even to increase that further to get these titers up, to get to protection from infection. And this is just a correlation between, on the left side, the saliva IgG and the serum IgG, which, there's a nice correlation because the assumption is that the saliva IgG comes from serum. When we correlate the secretory IgA with serum IgG, there's also a correlation, but it's a much weaker correlation. And that probably has to do with the fact that the secretory IgA is made locally. And then, of course, in fall of 2021, we started to give booster vaccinations. And the question was, what would happen if we do that, right? What you see here is basically one subject from the group, where you have these 2 shots initially. This was a naive subject. You see this increase in antibody titer, you see waning, stabilization, then the person got the booster. And the question is now, how far down are the titers going to fall? Are they going to fall down to this level, or are they going to stay up higher? And so we looked at that. On the left side is the same graph that I showed you before. This is after the first 2 shots. After booster is shown on the right side, and day 0 here is the day of the booster vaccination. And again, before booster vaccination, you have your 2 groups, the orange ones and the blue ones, and they're still separated. But then people get that booster vaccination, and that is kind of the unifier in terms of the titer in serum. And that's the first thing that stands out, that both groups now have similarly high titers. And the other thing that stands out here is that the kinetics are different, right? The waning is different. And don't ask me how we modeled that. We have 2 people that are shared in the department that do modeling, but they tried to model the decay kinetics that we see. Again, in blue are the naive individuals who got vaccinated twice. In orange are the hybrid immune ones that got vaccinated twice. And then in pink here, we have the waning after the booster dose for both. And what you see in the end is that there's less waning with the booster dose. And of course, the stabilization phase happens at a much higher level. And so we also started to look into what happens after the next booster and the next booster and the next booster. And basically, we don't get up to a higher baseline level, but it stays relatively higher. The other question was when the bivalent booster was rolled out. And the question here was, would we make any antibodies that are specific to the BA.5 that's in the bivalent vaccine, right? And this is what I'm trying to show you here. First, this is sera taken from people who got the bivalent vaccine pre and post vaccination. Here on the left side, you have binding either to the wild type receptor binding domain or the BA.5 receptor binding domain. And in both cases, the binding goes up, right? Here we have neutralization of wild type and BA.5 virus. Again, in both cases, the neutralization goes up. And here on the right side, you see avidity. And in both cases, the avidity goes up as well. But that doesn't solve the question if there are specific antibodies to BA.5 that are induced by that vaccine. So we did another experiment where we took all these sera pre-vaccination, post-vaccination, and then we used receptor binding domain protein from wild type, from the ancestral virus, to pull out all the antibodies that would bind to the wild type RBD. Okay, this is shown here. So here you have ELISA titers pre and post vaccination against the wild type RBD, and the dashed lines here are the titers that you get when you pull all the RBD out, antibody binding antibodies out of the sera, right? So you can completely remove that signal. If there's any antibody present in that sera that binds to BA.5 RBD specifically, there should still be reactivity now against BA.5, right? And so we did that experiment. Here's BA.5 binding shown, again with the undepleted sera. There's binding, of course. And then with the wild type RBD depleted sera, and there's no binding. And that indicates that at least with the sensitivity that we have here, we cannot detect any BA.5 specific antibodies after the bivalent vaccine in sera. This doesn't mean it's bad, because there's a lot of cross-reactive antibodies that are highly neutralizing and they are protective, right? But there's nothing specific. And this goes along very, very well with what Ali El-Abadi has seen at Washington University in the memory B cell compartment. They see very, very few memory B cells, 1 in 100,000 or so, that actually are specific for this BA.5 variant now. All right, I'm going to switch gears a little bit and I'm going to speed up. So we also wanted to look at the monoclonal response after vaccination. So we took the first 6 people in the cohort who got the mRNA vaccine. They were naive before vaccination. And we collaborated with Ali El-Abadi at Washington University to sort out monoclonal antibodies. And in order to do that, we sorted these plasmablasts that come up in the beginning and make antibody. And you don't need to sort for them in an antigen-specific manner, right? Because they are a very special population. You can sort them based on their phenotype, which means you can do that in an unbiased way. So we did that. We did a lot of serology, which I don't have time to go through right now, so we'll skip that. But we got a ton of monoclonal antibodies from 3 of these subjects, right? And so the antibodies are shown in these different lines here. And in A, you see spike binding of each of these different monoclonal antibodies, right? This is after vaccination with mRNA vaccine. But we also wanted to know to which parts of the virus these antibodies bind. And it turns out that about 1/3 of them bind to the receptor binding domain, another 1/3 to the N-terminal domain, and another 1/3 binds to S2, which is the lower part of the spike protein. And of course, when you have these antibodies, the question is, are they functional? Do they do something, right? So we ran neutralization assays with them, and one surprise was that actually very few of them are neutralizing. In this first subject here, we found 1 neutralizer that bound to the RBD. In the second subject, we found 1 neutralizer that bound to the NTD. And then in the last subject, that was the lucky one, we found 5 neutralizers. One of them bound to the RBD. But that left a lot of antibodies that were non-neutralizing. Some of them were cross-reactive. I'll skip that part. But we got all these non-neutralizing antibodies. And so the big question was, what are they doing, right? Are they beneficial? Are they detrimental? Are they just dead weight and they are induced, but they don't do anything? And in order to test that, we went back to a mouse model, right? So we passively transferred these antibodies into mice, and then 2 hours later, we challenged them with a mouse-adapted version of SARS-CoV-2 from Ralph Barick's lab. And then if the mice get sick, if they're not protected, they lose weight, and they might cross a humane endpoint, and then you have to basically score them dead. And so we did that with all these antibodies that we found, including the non-neutralizing ones. And here are some of the results. We have much more data on that by now, so about the mechanism, but it was surprising in the beginning. What I'm showing you here is RBD antibodies in A, anti-D antibodies in B, S2 antibodies in C, and then we had some where we couldn't figure out the epitope. Those are shown in D. And the neutralizers are in red. And what you see here is weight loss in mice. So if the line doesn't go down, that means protection. And we see that the neutralizing antibodies actually provide nice protection in the mouse model. That makes sense. They neutralize the virus that protects the mice. But actually, a good number of the non-neutralizing antibodies also provided protection. I don't have time to go into that right now, but this is probably because of interactions between the Fc part of the antibody and the Fc receptor and effector cells and clearance of infected cells. And the cool thing is a lot of these antibodies that are protective here are actually cross-reactive, even to other beta coronaviruses. And why do I think this is important? When Omicron came around, right? When people looked at the sequence, it became very clear that most of the neutralizing epitopes had changed, and the expectation was that neutralizing antibodies against Omicron would be much lower as compared to the variants before that. And this is just shown here from one of our studies, where you see, for example, here convalescent individuals and their neutralizing antibody titers against wild type. And then in red here we have Omicron, right? So about 75% of them lost any neutralizing activity. And even for vaccinated, triple vaccinated or hybrid immune individuals, there was a steep drop-off, often 20 to 25-fold. But at the same time, the binding antibody was maintained to a large degree, right? The drop-off there was about 2 to 2.5-fold. And we think this observation that Omicron basically, while there's not good protection anymore against infection in the population, rarely causes severe disease. It's probably A, because there's cross-reactive T cells that protect you from progression to severe disease. But we also think that these non-neutralizing antibodies play a role in that protection from progression to severe disease. They cannot protect you from infection because they don't neutralize the virus, but they can probably clear infected cells, and that could be very helpful in this context. And so we saw that, and the next question is, how far does this reactivity go, right? And so this is a phylogenetic tree of coronavirus spikes. Here, this blown up part, these are all beta coronavirus spikes. Here we have the Sarbecoviruses, we have P-Becos, No-Becos, Mer-Becos, M-Becos. And then here we have a few alpha coronavirus spikes, and then gamma and delta coronavirus spikes. And so we made actually all these spike proteins, right? I mean, there's of course many more. This is a very small selection, but we wanted to see how far on this tree cross-reactivity would go. So we made all these proteins, and this is again, ELISA reactivity. And so what you can see here on the right side is reactivity of pre-pandemic sera to all these spikes. So the different spikes are here on the x-axis, reactivity is shown on the y-axis, and it's pretty clean. We don't see much before the pandemic, except for the alpha coronaviruses and the beta coronaviruses that circulate in humans, right? And you would expect reactivity there. That includes OC43 and HKU1 for the beta coronaviruses, and 229E and NL63 for the alpha coronaviruses. And then there's reactivity to the bovine coronavirus here, which is pretty strong. And that also makes sense because that's closely related to OC43. The idea is OC43 branched off from bovine coronavirus and jumped into humans. So this makes sense. When you look after SARS-CoV-2 infection, this starts to look very different, right? We see low titers, but we see consistently titers against a lot of these beta coronaviruses in the population now. Given this is shortly after infection, right? Titers are high, but you see this reactivity, and there's a proportion of people that even respond to gamma and delta and gamma coronavirus spikes here. And when you look at people who had 2 or 3 shots of mRNA vaccine, this includes now hybrid immune individuals, you see that there is actually a strong induction of antibodies to all subbacoviruses. And here, this is Omicron, right? And this is SARS coronavirus 1. The response here is almost the same. In fact, we do detect neutralizing antibodies against SARS-CoV-1 to a certain level, not very high, but we do detect them. But even these other subbacoviruses, which include viruses that have unknown receptors, this one, for example, was found in bats in Bulgaria. Even there, we have relatively good reactivity. But we have okay reactivity across the beta coronaviruses in general, and this extends to gamma and delta coronaviruses as well. And this is super interesting. These different cross-reactivity profiles might be caused by different antibodies. We know that across beta coronaviruses, we can have S2 binding and cross S2 binding. For the gammas and deltas, it's probably binding to the fusion peptide because that's highly conserved. We don't know exactly what this means, but we think that we're in a very different situation in terms of immunity to coronaviruses as after the pandemic as compared to before the pandemic. And maybe that also means that there's more resistance in terms of severe disease to a new coronavirus that would jump into the human population. This is speculation at this point, but it looks very different. All right, I have very little time. Do you want to hear a vaccine story about vaccines that we are developing, or do you want to hear about antibodies that are induced by mRNA vaccines for which we don't have a function explanation, but which are very weird? Our vaccine, okay. All right, so then I'm going to go through that last slide. So this started with an interesting story, right? In the beginning of the pandemic, we did not know which vaccine platform would work, and we did not know which vaccines could be produced at large quantities. For mRNA, this had never been done. It was unclear if you can make millions of doses, right? And so in our department, we never stopped working, right? But certain PIs who were above a certain age were asked to go home and work from home, right? And so I remember at some point I get a call from Peter Balesi, the department chair, that he wants to meet with me and Adolfo Garcia Sastre because he has an idea for a COVID-19 vaccine. It was very sketchy because Peter was at home, Adolfo and I were at work. And so we decided that we would meet. It was dark, it was winter, of course, right? In Central Park at the playground. And so there were these masked weird virologists standing there talking about the vaccine idea. And Peter was like, we need to use the global production capacity for flu vaccines to make COVID vaccines. I think he thought that's a good idea because you could reuse that, right? And so he thought about that and he found a way to do that. And this is what I'm showing you here. So basically, we based the vaccine on Newcastle disease virus, which you might not have heard of. That's a virus that is a problem in poultry, infects chickens. There's a highly pathogenic version that kills chickens very quickly. It's non-dangerous for humans. It can deal with our innate immune system. But because it can infect chickens, a vaccine strain was made. There's actually several. One of them is the Lesotho strain. So that's safe for humans and for chickens. But because it's a chicken virus, it grows very well in embryonated eggs, yeah? And embryonated eggs is exactly what is used to make flu vaccines. So we decided to engineer this virus and in addition to its own glycoprotein, give it a version of the spike protein of SARS-CoV-2. That spike protein is modified, it's super stable, it's non-functional. It was stabilized with the constructs or with the mutations that Jason McClellan had developed at the University of Texas, Austin. So it's a hexapro spike. And then we engineered it so that it would have the transmembrane domain and the cytoplasmic tail of the F protein of NDV. And what that did was, it was very nicely incorporated into the Newcastle disease virus virions, right? And so you end up with a virus that basically has a lot of spike on the surface, super stable spike. And if it goes into a mammalian cell, it starts to express its genes, including the spike, and then it's getting shut down by the interferon response. And so there's a lot of spike present plus interferon, and interferon has an adjuvant activity, right? So it's actually a nice vaccine vector. And just to show you that we really get integration of the spike into this virion. Here we have preparations of wild-type Newcastle disease virus on an SDS page, and you see all these bands, which are the NDV proteins. And here on the right side, we have our NDV hexapro-S, that's how we call the vaccine, and that has an additional band here. And that's the spike protein. It's the pre-fusion uncleaved spike protein. And as I said, you can now grow this virus in X. It actually grows better there than flu. And if you want to make a vaccine, you can do that in two ways. The same we do it with flu. You can either grow the virus and then not inactivate it, and you can use that as live vaccine. And that also allows you to use it as nasal spray and potentially get mucosal immunity. Or you can inactivate it and then give it intramuscularly, also like an inactivated flu vaccine. And so for both of these processes, there's plenty of production capacity worldwide. Of course, we tested all of this in animals initially. I'm not going to go through that animal data. Just one key data set where we used the live version as a nasal vaccine in hamsters, which are a really good model for SARS-CoV-2. We had 4 groups. The group 1 got intranasally vaccinated twice. Group 2 was a control that only got the wild type Newcastle disease virus without the spike. Group 3 only got BBS as control, and then group 4 was a healthy control. There was no treatment of those animals. And we see that when we vaccinate the animals, we get a lot of serum IgG, we get neutralizing activity. But the most important thing is when we challenge these animals, when we infect them with SARS-CoV-2, they look like the healthy controls, the group that did not get infected, right? The healthy controls are here, the dashed line, and in blue, we have our vaccinated hamsters. So there's basically absolutely no disease in that model. And we also see basically no replication of virus in these vaccinated animals in the lung, and very little to no replication in the upper respiratory tract. And that's important. So they make a mucosal immune response that really controls the virus very well. And so these vaccines actually moved forward into the clinic. The first 3 are 3 line items that you see. There are collaborations with GPO in Thailand, IVAC in Vietnam, and Instituto Butantan in Brazil. And we just gave them the vaccine constructs, and they make inactivated vaccines, and then they started clinical trials locally. This was done under humanitarian license, so we just gave them the material, and they started going. And at least I think we have to shut down soon. At least in Thailand, the phase 3 trial for this is now fully enrolled. In Mexico, the phase 3 trial, we did something similar with a company in Mexico, although they use a live version of the virus, and that's now fully enrolled in phase 3 too. And at Mount Sinai, we tested the nasal version, and we're a little bit slow at Mount Sinai. We're just done with the phase 1 trial now, but it looks very promising, and we do see an induction of mucosal immunity in humans as well. I think I'm going to wrap up because I've heard that students are going to come into the room in a second. But basically, we think we have a relatively nice vaccine strategy that could work, right? It needs to be tested, and we need to figure out if it really protects in a phase 3 trial from infection, but it's at least worth testing. And I think I'll stop with that. Sorry for running out of time.
Thank you so much for this fantastic talk. Immunology is always fascinating. Unfortunately, we don't have time to take questions, but I'm sure if you reach out to Florian, he will take his 25-minute flight to answer your questions when he fly back to New York City. All right, I think I'm going to wrap up because this is our last seminar of this year. What? Oh, okay. Okay, one question on site. Okay, the last row.
Curious if you're aware of any phenotypic or clonal signatures of the B cells in the people that had boosted mucosal after vaccination.
That's a good question. We haven't looked into that. We have the BVMCs. We could do that. We of course don't have local B cells, but we could look into that. We could look into homing markers. That's certainly something possible. We haven't gotten around to do that yet, but the samples are banked.
Okay. Yeah.
Yeah, we can do that too. Yeah, please.
What I was really attracted to the graph that you showed about the model, where you have I think no infection by vaccination compared to infection and vaccination. Looks like that was a drastic difference. What do you think will be the key hypothesis behind that difference?
I mean, to be honest, I think it might be just a number of factors that come together. It could be the mucosal component that plays a role. It could be just the quantity of antibody that these people have. They have much more than the people who were, just vaccinated. But it could also have to do with the number of exposures, right? You're talking about 3 exposures versus 2. And while the second dose of the vaccine didn't seem to increase the titer, it might have stimulated affinity maturation even more, right? So I think there are differences there that could drive that. To pin it down to just one is difficult. However, there are studies by Jen Gommerman in Toronto and Charlotte Dahlin in Sweden that do suggest that the question of breakthrough infection versus no breakthrough infection can be pinned to mucosal immunity to a large degree. So those studies are out there, but we don't have data that would allow us to say, oh, it's just mucosal immunity.
Brad, can you pass one more question from the online audience?
Do you think that the cross-coronavirus spike finding ADCs like long-lived targeting highly conserved epitopes, or are they also related to recency of infection? Does the level of cross-reactivity decrease over time? That is, do you think you might see a similar cross-reactivity if pre-pandemic samples were obtained soon after a seasonal coronavirus infection?
That's a super excellent question. I think it's both, right? I think the higher titers, they're not going to go away. So I think that stays. But those samples were taken weeks after infection, or 1 to 2 months after infection or vaccination. And so the titers are high. And of course, they drop over time, and some of them might become undetectable. But the memory B cells might still be there, right? So I don't think they will completely go away, and I don't think it will look like before the pandemic. But the really high titers that you see after 3 times vaccination, I don't think that will be maintained at that level.
We can take one more question. Yes, please.
And it brings to mind a little bit like the weird observation that BCG vaccination, despite pretty useless in adults against TBs, is actually good when injected locally for certain types of cancers. Has there been any reports coming out that are reporting like a decrease in the incidence of certain types of cancers or regression in certain types of untreated cancers in patients? Although that might be unethical if you use on cancer. But like, is there anything like that coming out after SARS-CoV, like after the pandemic, to suggest that this might be true post-vaccination as well?
That's a complicated question. But so that's not cancer is not my field at all, but I haven't heard anything about that, right? We do work on a lung cancer project, and as part of that, not my group, but groups involved study replication of SARS-CoV-2 in different lung cancer cells. And it seems in some it grows very, very well and it destroys them, right? But if that would in vivo lead to a remission, I'm not so sure about that. there could be an effect, but I'm not aware of any data that would suggest that.
Brett, is there any question online? Anyone? Mark.
Terrific. I'm curious, you mentioned that most of the alterations that produced, or a number of them that produced on crop, were at neutralizing sites. But you show, at least for disease, but maybe also for transmission, maybe that there are effects of non-neutralizing antibodies. How do you think about sort of selection for escape from non-neutralizing antibodies?
Yeah, that's a very good point. There might be mechanisms, right? But the thing is, we see even transmission before you get symptomatic, right? Typically, I would assume that these antibodies have a good effect a few days in, right? It's more like a T-cell response. It's not the antibody binds and you get an immediate phenotype. It's basically the antibody binds. You need to get immune cells to the site of infections. You need to get macrophages in large numbers, NK cells, if you want to have these effector cells interact with the FCs, right? And that takes a while. And at least in the animal models, we see that too. So on a, day 2 after infection, you don't see much of a decrease in titer. But then if you wait until day 4, for example, you see huge differences, right? That's when these antibodies act. And I think that that puts a different pressure on the virus just because it gets a chance to transmit before there's a real effect on replication. That's my take on it, but I'm pretty sure, but that would be a gain of function experiment, I guess. You could test in an animal model if you could drive that resistance, but you would need to get persistent infections.
We have one more question from our online audience. Alain Chuni, sorry if I pronounced your name wrong. So their question is, have there been significant studies that attempted to correlate antibody responses in conjunction with CD8 cellular responses with respect to repeat infections?
First of all, I'm not a T-cell person. People are trying to look into direct correlations and see if there is a protective effect of the T-cell responses in terms of breakthrough infections, right? But you run into the same issue. A T-cell cannot prevent an infection. A T-cell can only prevent infected cells from spreading more virus at a certain point, right? So I'm sure there's a mechanism in terms of stopping progression from severe disease. But in terms of just preventing breakthrough infections, I think for a T-cell that would be hard, except if you have very high levels of tissue resident memory T-cells in the upper respiratory tract. But they're usually not maintained for a very long time. But the other problem that we consistently face with T-cell analysis is sampling, right? And the analysis itself, if you look at correlates of protection for antibodies, super easy because the assays are cheap and you can run them in high throughput. And that's absolutely not true for T-cells. And I think that's why there's some data lacking there.
Thank you. We are done with questions. Awesome. So on behalf of the Center for Communicable Disease Dynamics and the Harvard Chen School of Public Health, let's thank Dr. Krammer Florian again. Dr. Florian Krammer again for joining us today and for the fantastic talk and discussions.